Introduction. Do you know, my father is suffering from diabetes. Oh, was your grandfather also diabetic? I don't know, but why do you want to know that? I wanted to know if your father has inherited diabetes from his parents. How can anyone inherit disease? Disease can be inherited by the transfer of genetic material from parents to their offsprings. Let me explain you in details.
Objectives. At the end of this lesson, you'll be able to understand the discovery of DNA as genetic material, understand the structure of polynucleotide chain, RNA and DNA, identify central dogma, understand the packaging of DNA in the nucleus, understand Griffith's transforming principle, understand Hershey and Chase experiment, identify the differences between DNA and RNA. Identification of DNA as genetic material. In 1869, Frederick Maescher identified DNA as an acidic material in the nucleus and named as nuclein. Maurice Wilkins and Rosalind Franklin produced X-ray crystallographic images of DNA. Erwin Chargaff reported that the ratio between adenine and thymine and guanine and cytosine are constant and equal to 1 in a double-stranded DNA. Using these propositions, James Watson and Francis Crick determined the double helical structure of DNA in 1953. Polypeptide chain. A polynucleotide chain can be formed in the following steps. A polypeptide chain has phosphate moiety at one free end of ribose sugar. This is known as 5' of polynucleotide chain. Other free end of ribose sugar has attached OH group. This is called 3' of polynucleotide chain. Structure of DNA. DNA is made up of 2 polynucleotide chains with sugar phosphate backbone. The 2 polymers runs antiparallel, that is, one polymer has 5' to 3' polarity, while the other polymer has 3' to 5' polarity. The 2 polymers show complementary base pairing, where base pairs are bonded with hydrogen bonds. The 2 chains are coiled in a right-handed fashion. Bases are paired in the helix after every 0.34 nanometers and the helix turns after every 3.4 nanometers. There are roughly 10 base pairs in each turn of helix. The plane of one base pair stacks over the other in double helix. Central dogma. Central dogma refers to the flow of genetic information in the following manner. It was proposed by Francis Crick. Packaging of DNA helix in prokaryotes. A typical mammalian cell DNA double helix has a length of about 2.2 meters. How is it accumulated in a small nucleus? DNA is a prokaryotic cell. In prokaryotes, DNA is organized in large loops and is held by proteins in nucleoids. Packaging of DNA helix in eukaryotes. In eukaryotic nucleus, histones are organized to form a unit of 8 molecules called histone octamer. DNA negatively charged gets wrapped around the octamer to form nucleosome. Repeating units of nucleosomes form thread-like structures in the nucleus called chromatin. The nucleosomes in chromatin are seen as beads on string under electron microscope. Chromatin is packaged to form chromatin fibers. During metaphase stage, chromatin fibers get condensed to form chromosomes. This condensation requires a set of proteins called non-histone chromosomal proteins. Griffith's transforming principle. The discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. Frederick Griffith worked on two strains, pathogenic and non-pathogenic, of Streptococcus pneumoniae. Griffith found that when he mixed the heat-killed remains of the pathogenic S strain with living cells of non-pathogenic R strain, some of these cells became pathogenic. Griffith called this phenomenon as transformation. Biochemical characterization of transforming principle. Colin MacLeod and Macklin McCarty worked to determine the biochemical nature of Griffith's transforming principle. Hershey-Chase experiment. In 1952, Alfred Hershey and Martha Chase performed experiments showing that DNA is the genetic material of a bacteriophage known as T2. They grew some viruses on a medium containing radioactive phosphorus, producing radioactive DNA, and some others on radioactive sulfur, containing medium producing radioactive proteins. Radioactive phages were allowed to attach to E. coli bacteria. It was found that bacteria which was infected with viruses that had radioactive DNA were radioactive, while radioactive protein-infected viruses were not, indicating that DNA was the material that passed from the virus to the bacteria. Properties of genetic material. A molecule must fulfill the following criteria to act as a genetic material. It should be able to undergo replication. It should be chemically and structurally stable. It should provide scope for mutation. It should be able to express itself in the form of Mendelian characters. RNA. RNA was the first genetic material. Essential life processes and protein synthesizing machinery has evolved around RNA. RNA can act as a genetic material as well as a catalyst. RNA being a catalyst was reactive and unstable. Thus, DNA evolved as a stable genetic material from RNA. Which is a better genetic material, RNA or DNA? These are some characteristics of RNA and DNA. Thus, RNA is better for transmission of genetic information. However, DNA being more stable is preferred for the storage of genetic information. Did you know DNA can be synthesized in test tubes using a small amount of performed DNA, DNA polymerase, and all the 4 deoxynucleotides? This helps in purification of DNA polymerase along with the production of biologically active DNA. Summary. Let us summarize what we have learned. DNA stores genetic information. RNA mostly helps in transfer and expression of information. DNA being chemically and structurally more stable is a better genetic material. DNA is made up of 2 polypeptide chains. The base pairs in the DNA helix are attached by hydrogen bonding. Complementary base pairing occurs in the DNA helix. Frederick Griffith experimentally worked out the transformation principle.
Introduction. Would you like to have such hair growth on your face? Oh no, not at all.
How does our body decide how much hair growth and other features should appear in our body?
Genes decide the expression of characteristics in our body.
Correct. However, only some genes are expressed and others are suppressed. Why?
Some genes are suppressed to prevent the expression of abnormal features in our body, such as excessive facial hair growth.
You are right. But who decides whether a particular gene is to be expressed or not? Okay. So let us discuss today how are the genes regulated in our body. Objectives. At the end of this lesson, you'll be able to understand gene regulation in eukaryotes and prokaryotes, understand the functioning of lac operon, identify the goals and features of Human Genome Project, and understand the concept of DNA fingerprinting. Regulation of gene expression. Gene expression is the process by which information from a gene is used in a synthesis of a functional gene product. In eukaryotes, gene expression can be regulated at multiple levels such as transcriptional level, formation of primary transcript, processing level, regulating of splicing, transport of mRNA from nucleus to the cytoplasm, translational level. Positive and negative regulation. At transcription level, the activity of RNA polymerase at a given promoter is regulated by interaction with accessory proteins which affects its ability to recognize start sites. These regulatory proteins interact with sequences termed as operators adjacent to promoter. Such interaction can be both positive regulation, activators, negative regulation, repressors. Lac operon. An operon is a group of genes that are transcribed at the same time, usually controlling an important biochemical process. Lac operon. Francois Jacob and Jake Monod were the first to elucidate lac operon. It is the operon which controls lactose metabolism. It involves 3 genes involved in the processing of lactose. Z gene. It codes for beta-galactosidase that hydrolyzes lactose into glucose and galactose. Y gene. It codes for permease that increases the permeability of cell for beta-galactosidase. A gene. It codes for trans-acetylase. It consists of 1 regulatory gene, I gene, that codes for the repressor of lac operon. Regulation of lac operon. Lactose is the substrate for beta-galactosidase and it regulates switching on and off of the operon. When lactose is absent, the repressor protein is synthesized all the time from the I gene. In the absence of lactose, repressor protein binds to the operator region and prevents RNA polymerase from transcribing the operon. When lactose is present, lactose interacts with the repressor protein producing a conformational change in the protein, thus rendering it inactive. As a result, RNA polymerase gets access to the operator and begins transcription of the operon. Human Genome Project. The Human Genome Project, HGP, is an international scientific research project with the primary goal of determining the sequence of chemical base pairs which make up DNA and of identifying and mapping the genes of the human genome from both a physical and functional standpoint. It was started in the year 1990 and completed in 2003. Methodology of Human Genome Project. Human Genome Project was carried out by two approaches. By identifying all the genes that are expressed as RNA, expressed sequence tags, by sequencing the whole set of genome that contained all the coding and non-coding sequence and later assigning different regions in the sequence with functions, sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Such sequences are then cloned in suitable hosts, bacteria or yeast, using specialized vectors called BAC, bacterial artificial chromosomes, and YAC, yeast artificial chromosomes. These sequences were then arranged based on overlapping fragments using computer-based programs. Salient features of Human Genome Project. Human genome contains 3,164.7 million nucleotide bases. Average gene consists of 3,000 bases. Largest known human gene is dystrophin with 2.4 million bases. Almost 99.9% of the nucleotide bases are exactly the same in all people. The functions of about 50% of the genes are unknown with only less than 2% of genome coding for proteins. Chromosome 1 has most genes 2,968, while chromosome Y has least 231. There are about 1.4 million locations where single nucleotide polymorphism occurs in humans. DNA polymorphism. If an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. Such variations are observed mainly in non-coding DNA sequences because mutations in these sequences may not have any immediate impact in an individual's reproductive ability. As a result, these mutations keep on accumulating generation after generation and form one of the bases of variability. DNA fingerprinting. It is a technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. DNA fingerprinting involves identifying differences in specific DNA regions called as repetitive DNA. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and other small peaks are known as satellite DNA. Satellite DNA does not code for proteins, show high degree of polymorphism and forms the basis of DNA fingerprinting. DNA fingerprinting technique. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. It uses variable number of tandem repeats VNTR as a probe. VNTR is a mini-satellite DNA. It is a small DNA sequence arranged tandemly in many copy numbers that varies from chromosomes to chromosomes. The numbers of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. After hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA. Steps involved in DNA fingerprinting technique. DNA fingerprinting technique uses southern blot hybridization using radio-labeled VNTR as a probe. Its steps include A. Isolation of DNA. B. Digestion of DNA by restriction endonucleases. C. Separation of DNA fragments by electrophoresis. D. Blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. E. Hybridization using labeled VNTR probe. F. Detection of hybridized DNA fragments by autoradiography. Did you know? A multiple gene regulation system is a system that allows the simultaneous and quantitative regulation of many different genes in the same cell, tissue, or organism. Foreign genes in a cell can be regulated by the use of inducible promoters such as PR1A promoter. PR1A promoter extracted from tobacco is induced during the systematic acquired resistance response following pathogen attack. Summary. Let us summarize what we have learned. Regulation of transcription is the primary step for regulation of gene expression. Lac operon is the operon in bacteria which codes for genes responsible for metabolism of lactose. Human Genome Project was a mega project that aimed to sequence every base in human genome. Fingerprinting is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymorphism in DNA sequences.